The cell cycle process starts with interphase. Following interphase comes mitosis. Interphase includes G1, S, and G2. Mitosis includes prophase, metaphase, anaphase, telophase, and cytokinesis. The interphase process starts with G1, also known as GAP1, where the cell does most of its growing. The cells increase in size and synthesizes new protein and organelles. Following G1 comes S, which is the synthesis phase. New DNA gets synthesized as the chromosomes get replicated, and at the end of this phase, the cells have doubled the amount of DNA than it did in the beginning. After the S phase comes the G2 phase. The growth of the cell comes to an end when the cell duplicates the DNA in the nucleus. The cell prepares to divide, and the interphase is over. Now begins mitosis phase. Mitosis starts with prophase. Prophase is when the chromatin condenses in chromosomes. Chromosomes duplicate and become visible. Then the spindle starts to form and microtubules appear in the centromes. Then the nucleus disappears. After the prophase, the metaphase begins. During metaphase, the centrosomes line up across the middle of the cell and are attached to spindle fibers in the centromere. Following the metaphase comes the anaphase, where the sister chromatids separate and spindle fibers split. During telophase, the chromosomes gather at opposite ends of the cell. Two new nuclear envelopes will form, and all the organelles will appear. Last stage of the cell cycle is cytokinesis, when the two cells split. Let's watch the process one more time. When cancer is involved in the cell cycle, the cells duplicate much faster and uncontrollably because cancer cells do not respond to the signals that regulate the growth of most cells. The cancer-related genes are protoconn genes, which encourage cell division, and tumor suppressor genes, which suppress cell division. Cancer can be caused by smoking, chewing tobacco, radiation exposure, and viral infection, and much more. This is the duplication of cells in a span of 4 days. On the right side, there are regular cells, and on the left side, there are unregular cancer cells. As you can see, a tumor has already occurred in the left side.